faithfully convey value and interpret great books from around the world. Hello, this is Sally Talks Books. Today, the book we're discussing is Nano and Life. Let's start with the two key words in the title. First, life and life-related research have always been humanity's most pressing concerns. From developing new drugs to treat diseases to extending human lifespan, these are questions life science seeks to answer. As research deepens, we aim to understand life phenomena from more precise perspectives. This requires a new discipline. Nano. A nanometer is 1/1000th of a meter. A human hair is about 60,000 nanometers wide. Nanotechnology focuses on extremely small entities like molecules and atoms. By academic definition, studying objects between 0.1 to 100 nanometers falls under nanotechnology. So, how can nanotechnology advance life science research? That's the core focus of this book. The author, Sonia Contera, is an Oxford professor and co-director of their Medical Nanoscience Research Center. Sonia researches physics-biology-nanoscience intersections and regularly conducts public science outreach. Thus, her writing delivers authoritative, cutting-edge knowledge in this field. The book explores nanotechnology's impact through biomolecular research, disease treatments, and health management. We'll distill this into 3 sections. Part 1 examines why nanotechnology is essential for life research. Part 2 explores currently applied nanotechnologies. The final part will analyze 2 concrete examples of how nanotechnology treats diseases and enhances life quality. First published in 2016, this field has seen breakthroughs since then. So we'll include recent developments in this discussion. All right, let's follow the author into this tiny, mysterious nano world. First, to understand the mysteries of life, the author believes we must use nanotechnology, and there are 2 main reasons for this. The first point is that introducing nanotechnology is an inevitable trend in the development of life research. The second point is that nanotechnology can serve as a bridge for interdisciplinary collaboration. If we look back at the history of biology and medicine, we'll find that bringing these disciplines into the nanoscale is the general direction of technological advancement. In early history, doctors could only observe features like heartbeats and body temperature to form a superficial understanding of life. And after the Middle Ages, with the rise of anatomy, it sparked a wave of research on the specific functions of various organs. In the 17th century, the Dutch invented the microscope, allowing people to explore life's laws at the cellular level. And from the late 20th century, especially since the 21st century, with nanotechnology's help, we've finally entered life science's deeper, more fundamental realm, the world of biomolecules. The main molecules in living organisms include deoxyribonucleic acid, or DNA, and various proteins, among others. The current scientific consensus is that human birth, aging, sickness, and death all depend on the interactions of these biomolecules. These biomolecules are nanoscale in size. For example, DNA has a diameter of just 2 nanometers, while protein molecules range from a few to tens of nanometers, with a few being larger. For instance, a protein in the human body called titin can reach over 1,000 nanometers in length. And, however, our research on protein molecules mainly involves analyzing amino acid sequences and subtle spatial structures, which still requires nanoscale resolution and accuracy. So studying these biomolecules falls within nanotechnology's scope. If we can explore at the molecular level, it will greatly enhance our understanding of life science. The most typical example is the change in drug development. Using traditional methods, much exploration relies on human experience, essentially a process of trial and error. The general process involves first extracting components from natural plants, animals, or microorganisms, or synthesizing potentially useful drugs, then testing them on lab animals like mice to observe efficacy and side effects. This research process is inefficient and extremely costly. Until the mid to late 20th century, Developing a new drug often took several years and could cost hundreds of millions in upfront investment. The reason is we didn't understand the specific mechanisms of how these drugs interact with molecules in living organisms. We didn't know how drugs react with receptors on cell surfaces after reaching cells, nor whether certain molecules might affect genetic material in the nucleus. There was no alternative. These molecules are simply too small. But with nanotechnology, we can now see and manipulate these biomolecules, enabling deeper research. Since entering the 21st century, drug development speed has increased dramatically. For example, in response to COVID-19, pharmaceutical companies developed the first vaccine within 11 months. By contrast, in the 1960s, scientists took over 4 years to develop the mumps vaccine. This efficiency boost comes from our clear understanding of viral molecular structures, how viruses interact with human cells, and identifying key protein molecules, allowing targeted solutions and rapid vaccine design.
Another reason life science needs nanotechnology is that it enables scholars from other disciplines to participate.
Life is complex. Exploring it involves physics, chemistry, biology, and more. As the saying goes, different fields are like mountains apart. Before nanoscience, scholars from other fields struggled to enter life science without medical or biological knowledge. But in the last 20 to 30 years, many scholars from other fields have begun life science research through nanotechnology. They tackle life science challenges from interdisciplinary perspectives. For instance, chemists study how nanoscale molecules aggregate into macromolecules in saline solutions. Superficially, a chemistry experiment, this could actually help simulate protein formation patterns and unravel life's origins. Similarly, physicists studying nanoscale electrical discharges in materials can apply this to reading brainwaves, helping understand consciousness formation. Or material scientists have developed many tiny spheres, only tens of nanometers in diameter.
Later, these artificial spheres proved excellent as microcapsules for delivering specific drugs to treat cancer.
Even one of the most cutting-edge research fields today. Artificial intelligence is entering life science through nanotechnology. The changes in operational rules of biomolecules are extremely complex. Now we can use nanotechnology to accurately record information during biological system operations, including molecular size, movement, speed, microstructure, etc. Then by inputting this massive data into algorithms, we can simulate biomolecular changes using precise mathematical models. One of the most exciting areas here is using AI to predict protein structures. Proteins are essential components of all living organisms. Every human cell is filled with proteins, and understanding their precise structures is crucial for medical research. In recent years, with nanotechnology, we've observed some protein structures in detail. Building on this data, in 2021, an AI company under Google developed an algorithm that successfully predicted 350,000 protein structures. Professor Shi Yigong, a renowned Chinese scholar, called this one of humanity's most important scientific breakthroughs of the 21st century.
In summary, with nanotechnology as the foundation, research from physicists, chemists, biologists, and computer scientists converges.
With all this talk about nanotechnology, what specific technologies apply to life science? Broadly speaking, nanotechnology currently provides two key capabilities: observation and manipulation. First is observational capability. To study something, we must at least see it. Observing nanoscale objects isn't possible with ordinary microscopes. Common optical microscopes max out at 2,000 times magnification, sufficient for cells, but not for molecules like DNA and proteins inside them. Scholars have continually sought ways to explore at microscopic scales. For example, some developed equipment called X-ray diffractometers, like oversized cameras but using X-ray light sources. After X-rays pass through biomolecules, they form spots on photographic plates. Analyzing the position and shape of these spots, Scientists can deduce biomolecular information. The DNA double helix was discovered using this technique, but X-ray crystallography requires biological samples to be crystallized, a extremely difficult process that can take years. Hmm.
Progress in microscopic observation was slow until the 1980s, when a revolutionary tool emerged, the scanning tunneling microscope.
This microscope differs fundamentally from conventional multi-lens microscopes. Its key component is an ultrafine needle with a tip just 1 atom wide during operation. The tip approaches the sample surface, and by detecting electrical current changes, it accurately maps surface topography.
This achieves atomic-level precision.
While scanning tunneling microscopes easily image nanoscale objects, they're unsuitable for liquid-based research because they require electrical current measurements, impossible in liquids. Soon after, the atomic force microscope was invented, operating similarly with a sharp needle for nanoscale measurements. However, atomic force microscopes measure the weak force between tip and sample surface, using this to deduce surface contours. Thus, atomic force microscopes can directly observe molecules in liquids and even measure cell membranes and surface changes. Well, atomic force microscopy quickly became transformative in biomolecular research. One of its most famous applications is observing myosin in action. Myosin appears in nearly every human cell, participating in muscle contraction, cellular transport, and cell division, making it one of our most vital proteins. Resembling a two-headed snake, myosin measures only about 150 nanometers long. In 2010, with atomic force microscopy, scholars directly recorded dynamic images of myosin molecules in motion. During movement, myosin's two heads latch onto a protein microfilament, like a tiny cable car track, then advance along it. Meanwhile, myosin's tail adheres to materials needing transport, completing delivery. The entire process resembles a slender cart operating on monorail tracks. Utterly fascinating. Beyond observation, nanotechnology also enables molecular manipulation. Scanning, tunneling, and atomic force microscopes can not only detect but manipulate molecules, using their tips to push or pull individual molecules. With enough patience, one can even assemble molecules into desired configurations. Some precisely arrange thousands of atoms to create a 250-frame stop-motion animation titled A Boy and His Atom. This holds the Guinness World Record as the world's smallest movie. Beyond simple pushing or pulling, scientists fabricate materials with nanoscale precision. Manufacturing methods mainly divide into two categories: top-down approach and bottom-up approach. Top-down involves gradually etching larger objects down to nanoscale. Bottom-up creates special conditions to assemble atoms or molecules into desired shapes. A fascinating example is DNA origami, folding DNA into nanostructures. To understand this, let's revisit DNA composition. DNA contains 4 bases: adenine, guanine, cytosine, and thymine. They pair complementarily, A with T, C with G. Thus, arranging base sequences in one DNA strand allows binding with complementary strands. Scientists realized they could create DNA fragments acting as staples. Designed to complement specific regions of long DNA chains, these staples guide folding. When these bind, the small fragments fix positions along the DNA strand, folding it into desired shapes.
In 2006, scholar Paul Rothman used these DNA staples to bind long strands into a smiley face.
DNA origami has evolved. Now we can weave nanoscale boxes from DNA strands, like willow baskets. Even more fascinating, by controlling DNA strand bending, we can open or close the nanobox lid. What could such tiny boxes be used for? Scientists propose loading enzyme molecules inside them. Enzymes are catalytic proteins vital in food, pharma, and other industries.
Putting enzymes in DNA boxes would allow precise reaction control by opening or closing lids.
Ultimately, nanotechnology enables development at microscopic scales with immense potential. In Part 3, we'll explore how nanotechnology improves lives through drug delivery and organ transplantation. In 1963, first, Drug delivery scholars don't study getting drugs from pharmacies to you. They solve how drug molecules reach diseased cells, where nanotechnology excels. That is, using nanoparticles to navigate drug molecules. Before explaining techniques, let's examine medication problems patients face. Suppose a patient with inflammation takes an anti-inflammatory pill. After swallowing, the pill enters the stomach, breaks down, and active ingredients travel via bloodstream to the inflammation site. However, much of the drug gets absorbed by other organs during this journey. Ultimately, only a small fraction of drug molecules actually work. Thus, to achieve therapeutic effects, patients must take far higher doses than needed. For common medications, this issue isn't as severe. But in extreme cases like cancer, this overdosing can endanger health. The most typical example is cancer. Chemotherapy drugs primarily kill diseased cells. To ensure enough drugs reach tumors, patients must take extremely high doses. But these excess drug molecules also fatally threaten healthy cells. Thus, chemotherapy is agonizing, causing pain, diarrhea, hair loss, and even organ damage. So, can we deliver chemo drugs directly to tumors without absorption elsewhere? Yes, by leveraging nanoparticles' unique properties. In 1986, a Japanese researcher discovered nanoparticles accumulate around tumors. Tumors form dense but abnormal blood vessels with defective walls and loose cell alignment to access nutrients. Nanoparticles moving through blood easily accumulate at these defective sites. Think of normal vessels as smooth highways, while tumor vessels resemble bumpy dirt roads. Nanoparticles act like tiny cars easily stuck on these roads. This became known as the Enhanced Permeability and Retention (EPR) effect. Using EPR, scientists precisely deliver drugs to tumors. First, we create specialized nanoparticles. Designed in incisor shape to trigger EPR, they rapidly cluster around tumors. Chemo drug molecules are loaded inside or attached to these particles. Thus, nanoparticles become navigators, and drug molecules become bullets. These guided bullets precisely hit tumors without affecting healthy tissue. Challenges remain. Each organ's tumors have unique vascular networks. For different tumors, nanoparticles require specific sizes, shapes, and chemical properties, all controlled at nanoscale precision. That's 1/60,000th of a human hair's width. As you can imagine, this is an extremely challenging endeavor. Fortunately, growing numbers of researchers are contributing, and nanoengineers now have sufficient data to expertly control nanoparticle growth, improving drug delivery efficiency and accuracy. Today, many startups are exploring commercialization in this field. Other precision cancer approaches exist, like targeted therapies that recognize tumor-specific DNA mutations in cancer cells. These drugs only activate when encountering cells with those genetic markers. Designing such drugs relies on nanotechnology's precise biomolecular detection. Another nanotechnology application is in organ transplantation. Specifically, scientists use nanotechnology to grow compatible organs outside the body. Life constantly exposes us to various risks. From traffic accidents to amputations from infected injuries, organ damage occurs. Thus, repair becomes essential. Repairing disease or trauma-damaged organs remains medicine's urgent challenge. Whether severe burns or internal necrosis, transplantation is still the most effective solution for critically damaged organs. The core issue in transplantation is organ shortage. WHO reports a global 20-to-1 ratio of patients needing transplants to available donated organs. Only 1 in 20 patients receives a transplant. Factoring in rejection risks, successful cases are even rarer. This severe imbalance has spawned appalling organ black markets. To increase supply, growing organs in vitro is the most promising solution. If perfected, organ transplants could become as simple as replacing car parts. But culturing organs outside the body is extremely challenging, with one major difficulty being creating suitable environments for cell growth. Growing fully functional organs isn't just about providing nutrients to cells. In real human organs, cells arrange themselves in highly complex three-dimensional structures. Within these 3D structures exists a nanoscale network scaffold. This network scaffold is called the extracellular matrix. Think of ECM as scaffolding. Cells grow within its grids to form functional tissues or organs. ECM composition is relatively simple, mainly proteins and carbohydrates. Collagen, which we often hear about, is one ECM component. But ECMO scaffold structures are highly complex, with each organ's network having unique characteristics. Scaffolds vary, some softer and stiffer, some with larger and smaller pores. To grow organs in vitro, we must mimic these networks, requiring two key challenges. First, measure detailed scaffold parameters in biological organs. Second, accurately manufacture artificial scaffolds matching those parameters. Nanotechnology plays a crucial role in both steps. For parameter measurement, before nanotechnology, probing live tissues was extremely difficult, let alone at ECM's microscopic scale.
But atomic force microscopy now precisely measures parameters like elasticity, viscosity, and porosity of cells and scaffolds.
With this data, scientists gain clarity on designing artificial scaffolds for specific organs.
Manufacturing these scaffolds also requires nanotechnology, controlling structures at nanoscale precision to accurately mimic ECM networks. Several nanotechnologies now exist for creating artificial scaffolds. For example, electrospinning can weave polymer fibers, just tens of nanometers thick, into specified networks.
The whole process resembles weaving a nanoscale fishing net.
Additionally, we can control the growth of hydroxyapatite material to form nanoporous structures. This material and its porous structure are ideal for bone growth substrates, and hydroxyapatite has excellent biocompatibility, meaning minimal rejection when implanted. As research advances, scientists have made breakthroughs. Tissues like skin and cartilage can now be mass-produced. But kidneys, livers, and hearts present greater challenges and value in artificial organ development. These organs feature extremely complex vasculature and tissue details, making progress slow with scaffold-based methods alone. To create these organs externally, nanoscientists proposed a radical solution: 3D printing entire organs. Regular printers use ink for flat surfaces. 3D printers layer materials to construct three-dimensional shapes directly. Recently, 3D printing has expanded to plastic toys and metal components. In 2019, Tel Aviv University researchers 3D printed a heart using patients' own cells, causing a global sensation. Scientists first induced patient stem cells into cardiomyocytes, heart muscles, and endothelial cells, blood vessels, then processed collagen and glycoproteins into a gel-like extracellular matrix. If cells are building bricks, this ECM gel acts as binding cement, all becoming three-dimensional printing materials. Simultaneously, a high-precision digital heart model is created via algorithms, even mapping capillary networks. Guided by this model, a specialized bioprinter then prints the organ, heart. Though this heart measured just 2.5 centimeters, similar to a rabbit's, it contained fully formed cells, blood vessels, ventricles, and chambers. Best of all materials came from the patient. This completely avoids the troublesome rejection issues in organ transplants. Of course, 3D printed organs remain far from practical use. Just printing is just the first step. Making them function properly inside the body poses even greater challenges. The deeper we explore life, the more we marvel at its wonders. From visible tissues and organs to micron-scale cells, down to nanoscale proteins and genetic material, life reveals multi-layered complexity and beauty. In future explorations, nanotechnology will play an ever-growing role. But nano isn't science's boundary. We'll venture into more microscopic, mysterious realms to profoundly understand life and unravel its mysteries. So concludes Nano and Life. That wraps up our interpretation. Find study notes below the audio. See you next time.